Hello, everybody. I think it's about time. I think we've got a significant amount of people that have joined, so we can get started. Welcome, everybody, to the December 3rd NBSTRN National NBS Pilot Study Call. Let's see if my slides will work. There we go. I'm just going to go through a few housekeeping items before we get started. So if you don't mind, please mute yourself if you haven't already done so. When other people are speaking, we want this to be a free discussion format. So if you do have a question asked, feel free to unmute yourself. But if someone else is talking, please keep yourself on mute. We also have a chat window that's available too. So if you are want to ask a question, you can also keep it asking in the chat window and I will be monitoring that. This call is recorded. And if one of your colleagues missed it, feel free to let them know that it will be posted on the NBSTRN YouTube page. So this meeting is going to go just a little bit different than our previous meetings. We're going to be focusing on adding another condition. We're going to add Duchenne muscular dystrophy. We're going to have a discussion about current pilots going on for this disorder. And then hopefully we have time so we can check on the other pilots from other states that are performing it for the new conditions. And then we also have a few updates from APHL, New Steps, and NBSTRN. And the next call will be in January. So briefly, before I get into the discussion about Duchenne, I just want to remind everybody that NBSTRN has been having these webinars for, I guess, close to a decade now. We started off having pilots study calls for SCID when that was the new disorder that was just being added. And it's very different than the other disorders that were previously newborn screening programs were added. So we had the discussion about the screening methods, the follow-up. And then after a lot of states had added SCID, we transitioned to talk about other conditions that were newly recommended as well as candidate conditions. So we just want to continue to expand this call to add other conditions of interest. This month, we're going to be talking about Duchenne muscular dystrophy, but in the future, we might be talking about other candidates or new developments in newborn screening. So if you guys have any conditions that you would like to talk about in a future call, feel free to email me, or you can, or Amy Brower, and just shoot us a note and we're open to discussing another condition. But before we start, I'm just going to give a brief background about Duchenne. I know most people on this call has heard of this disorder. It's been around for a while. In fact, on the slide, I have the first pilots were started in the 70s using creatine kinase assays. But just to remind everybody, Duchenne muscular dystrophy is an X-linked neuromuscular disorder. The incidence is estimated to affect 1 in 5,000 males. However, it does affect females too in rare occasions. It's caused by deletions, duplications, and variations in the dystrophin gene. There's several treatments that are already available, and there's also more treatments in the pipeline. And we choose this because there's several pilots that are going on today because there's currently an FDA-approved screening assay from Perkinel to detect this specific isoform, creatine kinase, the MM isoform. So we're going to start the discussion today, hearing from three different pilots, one from New York State, then Early Check, as well as Wisconsin did a validation. We're going to talk with these, but if someone else has something to add, feel free to chime in and give your input. So with that said, I think I saw Michelle, she's on the line.
I am. How are you?
Good, how are you? You want me to, I'm going to stop sharing my screen.
Okay, and then I can.
You should be, if not, I can make you the host.
I have two monitors, so let's see. Can people see this?
We see your one. Yeah, we can see your slides up now.
Okay, great. Let's do not that. Huh, I never had this version before. Okay, well, can you just see the slides and I just see my notes, right?
We see your
look
slides
different on
and your
both,
notes,
so.
but that's okay.
Okay. Thank you. Thanks everybody for joining, and good morning or good afternoon, depending on where you are. I'm gonna go over the slides that were presented from the APHL meeting. This work was presented by Norma Tavicoli, and these are adaptation of the slides that she used. So we'll get started really quickly, and I'll try not to take up too much of your time. I should start by saying the pilot study is funded by the various companies that you see listed on this slide, and we also had a lot of support from PPMD, obviously, and PerkinElmer to do the study. So as Jennifer said, the Duchenne muscular dystrophy is an X-linked recessive neuromuscular disorder. The quoted incidence is about 1 in 5,000 males, but due to skewing in X inactivation, some females can also have manifestations of Duchenne. It's somewhat of it could be a candidate for newborn screening, primarily because the symptoms don't occur right at birth. They occur a little bit later on, and they're usually by phenotype. And the mean age of diagnosis still is somewhere around 5 years of age. And the symptoms, I won't read through them all, are listed here, but most people associate weakness and difficulty in walking as time goes on. Children generally become wheelchair bound, and while life expectancy has improved, still individuals that are affected with Duchenne live somewhere to the early 30s. And so the idea is if we can treat these boys and some girls early, that life expectancy can become normal is the ultimate goal, obviously. The dystrophin gene has mostly large deletions and duplications. There's also point mutations, and depending on where in the gene the variants occur, you can have Duchenne or a more mild form called Becker muscular dystrophy. Dystrophin molecular testing is a little bit complex. It's the largest known human gene. It has 2.2 million base pairs, 79 exons, and 8 different promoters. And as I mentioned, most commonly, the mutations are deletions in specific regions. And some of the treatments have been targeted to that, to fix that X and skip those exons so that a functional dystrophin could be generated and the protein would become functional. So one of the early treatments is corticosteroids, and then there's been several drugs that have been approved for the treatment of Duchenne. And of interest, there's a lot of additional treatments that are in the pipeline with the goal of some of them actually being curative with respect to gene therapy or gene replacement, and at least, if not cured, at least drastically improving muscle growth and function and reducing inflammation. So as Jennifer mentioned, there is an FDA-approved assay that PerkinElmer developed and is run on the GSP instrument, which is a common instrument in newborn screening laboratories. And this is targeted towards the creatine kinase MM form, which is the muscular form. And the idea behind the screen is that babies who have Duchenne, even though they're asymptomatic at birth, will have elevations in the creatine kinase MM isoform. But people have to also understand that this is an indirect marker of Duchenne. It's really a global marker of skeletal damage, skeletal muscle damage. And so you can also have elevations in CKMM from other conditions, and also different birth incidents as well. So in order to improve the specificity, if you're interested in really targeting Duchenne and those kids, you have to include, at least right now, some other second-tier tests, whether it's molecular at this point or other biomarkers that come into practice as more and more people begin to investigate screening for this condition. As Jennifer also mentioned, there's been internationally, there's been several different screening programs that have implemented screening for Duchenne, and a lot of these were done before the CKMM isoform was available, the testing was available. And probably the most famous one is the Ohio pilot study, which was done by Jerry Mandel and colleagues back in the early 2000s. So we decided, along with PPMD, to implement a pilot project to look at newborn screening for Duchenne. There's a lot of people that are involved in this and a lot of players that are helping get this project going. Our program is doing the screening. PPMD runs the Duchenne Steering Committee. They also gathered the funding for this project, and we work with them pretty closely. Our project involves 2 health systems in New York City. We also work with NBS-TRN, PerkinElmer, and again, the funders. And we also work with a New York State permitted laboratory to do the 2nd-tier molecular testing. We are not doing that in our newborn screening program, at least yet. So the goals were first, obviously, to validate the 1st-tier immunoassay. When we first began this, PerkinElmer did not yet have FDA approval. So when we started the validation, we were using a non-FDA approved test and had to work through a validation process for that. Also optimizing a 2nd-tier strategy to perform the molecular testing and make sure that that was done consistently for babies who screened positive. The idea obviously was to find babies who were at high risk to develop Duchenne before they were actually displaying a phenotype. And the idea is that you would enable these parents to confirm the diagnosis early. They could participate in clinical trials and be in a better place to determine a treatment, a mode of treatment for their child. The other goal of this was to use the results of the pilot study to provide evidence for state and federal assessments of the benefit of screening for Duchenne, create the infrastructure and educational materials and long-term follow-up with the NBS-TRN, and then obviously at the end of the day nominate Duchenne to the rest. And these are all in various stages of process. So because it's a consented pilot study, we developed a protocol, a video, a brochure, the consent forms, and SOPs. We had to obtain IRB approval at all the institutions, obtain equipment, reagents with the help of PerkinElmer. We hired someone at Newborn Screening, Sanju Park, who's doing the testing here, get all of the participating hospitals on board with their coordinators, validate the assay, and get CLEP approval to perform the test for newborns in New York. The validation, we did almost 9,000 babies tested blindly, and we also were able to get Duchenne cases, retrieve specimens from cases across the state in order to test their newborn screening specimen from birth, and got consent from parents to do that. The whole validation ran about 3 weeks. We validated 2 instruments, and obviously you need more than one person running the assay every day. And so we wanted to assess the variability in the assay and also factors that could influence the CKMM values you would get in a screening assay. And so the chart on the top left here just shows a scatter plot of all of the values from the validation, and you can see at the top that where the arrow is pointing, these cases up top here were from the specimens from babies who later went on to develop Duchenne, but most of the specimens cluster down in the 0 to about 2,000 range, and this just shows a frequency distribution of that validation data plotted out. 2 factors that we thought could influence the CKMM values were gender and also the time when the specimen was collected. While sex did not impact, males and females had roughly equal CKMM values, we did see changes based on when the sample was collected and how old the infant was at the time of collection. And so this led us to establish not necessarily gender, and we didn't see variation with birth weight either. So our cutoff values for the assay were based on that data I showed you, and we had different cutoffs based on the age at collection. We decided to establish a borderline cutoff where we would request a repeat specimen because as anytime you do one of these tests, you always have 1 or 2 samples from confirmed children with the condition who end up having the actual condition, and their values are lower than everyone else. And so we had 2 specimens where the boys had Duchenne and their values were a little above 2,000, just around, I think one was like 2,200. And so we set a borderline cutoff, and we decided to ask for a repeat specimen for any babies who fell in that range. So the screening strategy is shown here. The samples come to newborn screening. If you're below the cutoff, you're good to go and you're screen negative. If you're in that borderline category, we request a repeat. And then if you're above that referral level, based on how old you were when your sample was collected, you're referred to one of the two centers, either Dr. Chung at the Columbia Press system, or Dr. T. Gay and Dorota Gruber also at the Northwell system. And then Julia Nguyen works with Dr. Chung and Carrie Koval, our genetic counselors, and they talk to the family as well. So then we end up having the consult, and the samples are sent out for the second tier molecular testing. This just shows data from the first few thousand babies that we screened up through August 31st. The number of babies is about 14,000, and you can just run through this. There's really no difference in gender, which is interesting because both equal males and females, babies, parents of males and females did consent to do the screening, and the breakdown here of the age of collection. As I said, we did establish this borderline category, and for that data set, we had 118 babies that had borderline results, and we requested a repeat. Any baby who has their sample collected when they're less than 24 hours in our newborn screening program, we automatically request a repeat. And so their treatment because of a Duchenne borderline would be no different than if they had a specimen collected for any other newborn screening test. And this just shows you the different ranges of the demographics for these babies. Of the 118, we actually were successful in getting repeats for 102. If you notice the dates, we ran through the whole beginning of COVID. We're still in COVID, and we've had pretty good success in getting repeat specimens back. We had 101 babies whose samples actually resolved and there was 102 babies total that we received the repeat, and one baby had 2 borderlines, and from the screening algorithm, that baby becomes a referral after 2 borderline results. 6 families declined repeat, and 2 reasons we were given. Obviously, one was COVID, and the other was the mother was already a known Duchenne carrier. One family said they'd repeat the specimen later. One family did not return any calls, and the remaining 8 were expected to be received. Out of those 14,000-ish babies, we had 14 infants referred, so that's about a 1 in 1,000 referral. The breakdown in sex was 6 males and 8 females. And the CK values were just above 4,000, the cutoff, and then all the way up through 12,000, which is quite high. And it's kind of interesting because as we figured when we looked at this assay in the beginning, we would see not only Duchenne, but other variants and other conditions. And so our very first referral was delayed in follow-up. This baby is now in process again. Another family refused any follow-up after they originally agreed to do the testing. They didn't want to be bothered any longer. In total, we had 3 babies who were positive for Duchenne based on the molecular analysis. But if you just look through here, there's various other things that may impact muscle development, muscle damage in the other infants that were referred. And so even though we're screening for Duchenne, we're also making an impact possibly for other conditions. The second set of infants here, this family was interesting because they had a history of atypical adult onset Duchenne. So they were aware of the condition. Baby had a pretty high CK value. And again, we had an Alagille syndrome, some other variants and other myopathies were detected in some other children. And here's the last group. Again, you see, even though Duchenne's negative, we see variants possibly contributing to the elevated CKMM. So this could feed back to our program so that maybe we can change that borderline category or even get rid of it and make a determination whether it is useful for families to have this information. Obviously, you would think it would be, but that's something that we would need to look into further. So have been some challenges. Obviously, COVID really impacted our availability of testing. We went from in-person testing in April to remote recruitment, and that impacted the numbers that we were able to recruit. Also, delay in follow-up. Families did not want to come in for visits. They didn't want to come in for repeats initially. And there was a concern for a delay in repeat collection, but we really haven't seen that very much. And we need to continue to do some long-term follow-up on those babies. Consented pilots are staff intensive and they are expensive. There are other mechanisms, like we'll hear with Early Check, that may be useful for these types of pilots. We've had to work with coordinators and they work, 5 days a week, sort of business hours. So we're missing people on the weekends and after hour discharges. But because of COVID, we found that the remote recruitment actually works. And so our centers are working on trying to do the remote recruitment on the off hours after the mother goes home in order to be able to capture those babies, because by only working during business hours, obviously not all families could participate in the project. We spent a lot of time tracking and monitoring consents to make sure that we test all babies whose parents consented and we don't test babies whose parents did not consent. And also late consents when you're doing the remote. It's a lot of work to kind of go through and pull and make sure everyone we've accounted for everyone. A lot of exchange between the Columbia genetic counselors and Dorota and our staff here at the newborn screening program. The pilot study also requires changes to your limbs and all of that, and you have to align a pilot study with your newborn screening program. You want to report results out in a timely manner for all screening, and so you don't want your pilot to delay. There's different mechanisms to do that, and programs have to figure out what works best for them. And obviously staff turnover is always a challenge for programs and for hospital staff as well. So we think we've developed a successful approach to pilot newborn screening for Duchenne. We were able to validate the assay. It works quite well in our hands. We have 9 hospitals in the New York City area that are participating. We just onboarded another one, and we've screened almost 14,000 as of the end of the summer. And of those, we have 2 that are likely pathogenic outcomes for Duchenne, and 5 have other possible disorders. We plan to continue our pilot through the end of September in 2021, and to use all of the information that we're collecting to nominate Duchenne for the rest. And so the last slide is our acknowledgements. Dr. Tavakoli, who couldn't be on the call today, is the project director for us. Sanju, Brianne, and Kelly were from the newborn screening program. And they do the testing and all of the follow-up, all of the consent checking and all of that work. The Duchenne Steering Committee, we meet with them and they talk about the pilot changes we should make and give us their insight and contribute a lot of useful help to our project. PPMD staff, our staff at Northwell, Columbia, again, Every Life Foundation. There's a lot of people involved in this. And so I'm happy to answer any questions, and thanks for a few minutes to show this to you.
Thank you, Michelle. Very good presentation. So we'll open up for a couple minutes of questions, and then we're going to move on to Early Check. Does anybody have any burning questions right now?
I would love to, it's Kathy Swoboda. I would just love to say what an amazing study and what a phenomenal job during COVID. Just spectacular.
I agree. Did you learn Michelle, did you learn anything about the other conditions on your newborn screening panel? Did you see any elevations, say for Pompe disease, that might be, that had elevated CKMM as well, or no? Was it?
We did look. So we screened for GAMT deficiency, which we look at creatine and creatinine and guanidinoyl acetate. We do Pompe. We look more at the enzyme for that. So, but we wanted to take a look to see if we could sort of happenstance see a correlation, and the numbers are small, and so far nothing's like fallen out, and we've had some kids with fairly high CKMMs. So we have to go and take a better look at that, but on the really high ones that I said, hey, check this out, they they said that they weren't, they were basically normal. So I'm curious to see if that persists or go back and actually take a better look at that.
And then we have one question in the chat box. Great presentation. Thank you. Do you have information on the cost of treatment? So I guess I'm not sure in your slides if you discussed the outcome. Was there treatment that's already been offered to some of the patients that were true positives? And do you have any information on that, the type and the cost, I guess?
I don't know costs. I think is Julia or Dorota on? I thought I saw Julia. I really don't have costs for those treatments. They are antibody-based type treatments, or messenger RNA, sorry. I don't know. I'd have to actually check with the folks that do the treatment. I don't know if anybody from PPMD is on that might be able to answer that.
This is Nikki Armstrong at PPMD, so I'm on. I can't give you the cost off the top of my head for the treatments. I don't think either of the boys who are in this presentation that were diagnosed are eligible for the exon skipping based on their mutations. And in general, the corticosteroid treatments aren't typically started in infants. Although they could be, they're not usually. So yeah, Julia just commented they're not receiving treatment yet. So.
Yeah, I think the one was a exon 18, and the drugs are more in the exon 53, 51.
53 and yeah, and 51. Yep.
Any other questions? Well, thank you, Michelle. I think we're going to move on to the early check presentation.
Wait, I had one more question.
Okay. I'm sorry.
Sorry.
Go ahead.
I'm too slow of a typer. So I'm just wondering, is there a way to link? I mean, are you able to ensure that these babies, through the follow-up that they receive, get referred to at least the natural history studies that are ongoing, or at least potentially candidates for gene therapy going forward, or other things like that? Is there any sort of built-in ability to link them to studies? Or do you just leave that at the discretion of the clinic?
It's not built into the protocol per se. Certainly when they get counseling and they see the neuromuscular docs or the geneticists, that they would, be made aware of those. I know that there was the one that had a family history, and that seemed to be a later onset. So I'm not sure how, that goes, but we can't recommend as a program certain treatments per se. So it's really done between the family and the provider.
Okay, I was just curious if Wendy Chung or I can't remember who you said was doing, whether they had sort of a set protocol to kind of enroll them for future contact or sharing the information with other.
Yeah.
If there was some sort of mechanism that was done, I'm just curious.
Well, we are working with there's surveys and there's clinical assessment data that is being for the babies that the families that actually have babies who are affected with Duchenne. We have a whole forms built into MBS-TRN in order to put that data in,
okay.
The clinical type data, and so that would be captured in there as well.
Okay. Sounds good. Thank you.
Thanks again for the presentation. I think we're going to, for time, move on to early check. Is Holly PA on the call? I think I saw her on the call. Holly, are you muted? I muted. One moment. We'll give her 1 second. While we're giving her a second.
Okay, sorry about that.
Go ahead. Are you able to share your screen, Holly?
I am going to try.
Yep, we can see
Can you
it.
see it? All right, do you have the proper view?
Same view as, wait, she's getting it switched. Yep,
There
there.
you go. Great. Well, I appreciate having the opportunity to talk about Early Check and our recent edition of CKMM. And I also really appreciate speaking after Michelle because I was able to pull a couple of background slides out of my presentation. So this is excellent. So Early Check is a study that RTI developed a little over 2 years ago. We launched screening now, and it's a research study that is a partnership with our state academic institutions as well as the North Carolina State Laboratory of Public Health. And so this is a study, like I said, that we've been screening for a little bit over 2 years now. We started screening with SMA and Fragile X, and starting in November, actually initiated screening for CKMM as well. Similar, somewhat similar in many ways to the presentation you just heard of the New York pilot. This is also a consented research study, and the individuals who consent to this study actually consent to all 3 conditions currently on Early Check. So people don't pick and choose conditions, they get the whole package at this point. And the research study was designed to develop and evaluate methods to offer voluntary screening to parents in North Carolina. We have a birth rate of about 120,000 parents per year, and to acquire data to inform newborn screening policy. And you can see a couple things that we're particularly interested in looking at below, but for the sake of time, I'm not going to get into a whole lot of detail. Really, we did develop Early Check to be a foundation to start to add and subtract conditions as we need to obtain particular kinds of data about particular conditions, and envision a future in which states can offer a voluntary panel for non-RUSP conditions. We really are looking for a different model where there would be a state newborn screening model and then this voluntary panel as well, because there are many conditions that really probably don't well fit under mandated state newborn screening. Don Bailey, you see his picture there. He is the PI of the entire Early Check study, and I'm the PI of the CKMM sub-study, and I'll start telling you a bit about that. So the high-level objectives of adding CKMM screening to Early Check, the first and simplest component is to offer screening under our voluntary research protocol to parents in North Carolina. So really to make the CKMM screening available to all of those parents. And we use residual newborn screening blood spots to do our screening. So we don't require parents to actually obtain another sample. And that's one of the many benefits of our collaboration with our state laboratory. We also, of course, are attempting to obtain data that would facilitate transition of CKMM screening to standard newborn screening. And as part of that, to assess outcomes of the infants we identify as screen positive and the impact on parents and families. I'll talk a bit more about those as well. So the outcomes, specific outcomes, are contributing data related to the screening performance of the CKMM assay, to implement and obtain data on a protocol for diagnostic confirmation. You heard from Michelle some of the components related to diagnostic confirmation, given that CKMM is not specific only to Duchenne muscular dystrophy. So really aiming to figure out what other dystrophinopathies and other neuromuscular conditions that we might identify, as well as assess the rate of true false positives, depending on different cutoffs. And I'll show you our cutoffs in a moment. We are in process now of collecting short and long-term child assessment and family monitoring data, and are able to compare the data we get from CKMM screening to data from Fragile X and SMA and other conditions as we bring them on to Early Check, and to develop models and assess outcomes for clinical evaluation, genetic counseling, and then the transfer of these children to clinical care, which is, as Michelle indicated, and some of that question and answer started to get into, really one of the most complex and interesting components of these pilots is that transfer into clinical care. So we had go live for our CKMM screening on November 1st. The first week, we actually we consented and ran all of our babies who came into the Early Check system in October. So we actually have screening from October and November, and now we've just started December, so this is very new to us. Our second week, we began routine testing twice a week for the CKMM screening as part of the overall early check panel. So again, CKMM is one of 3 conditions or 3 analytes that are now being tested under the early check protocol. Our approach to confirmatory testing, I'll show you in a moment, I'll show you our cutoffs for the first and second screening test, but right now, and this is subject to change. I mean, I think Michelle mentioned this too. This is something that we plan to obtain data through February, and then using that set of data likely will change some of the approach based on having some of this evidence. But at this point, currently CKMM above 1626 is a positive screen. Once we have a positive screen, we initiate our telegenetic counseling protocol, which has already been in place for the other conditions. We relay the result to parents, and at that point consent for a next-gen sequencing panel that includes 86 genes that have a phenotype that has a impact on muscle and motor development. We also obtain a second CK value, and that's actually obtained at this point and run at LabCorp. So we have a second latent CK in infancy. And if necessary, we obtain a saliva kit sample for the next-gen sequencing, but the intention, and so far we've been able to do the next-gen sequencing based on residual blood spots as well. If there is a negative latent CK, we still have the next-gen sequencing run. And so if both of those are negative or uninformative, that would be a false positive result relayed to the parents. If there is a positive latent CK, we have a second round of communication that actually starts to integrate our neuromuscular provider to anticipate the potential for a diagnosis. And once there is a result back from the next-gen sequencing, then we actually start the whole cascade of follow-up. Our follow-up includes several rounds of genetic counseling and psychoeducational materials and psychosocial supports. We have a whole set of materials that are already, many of them developed and many more in development as we launch this study. They have a clinical assessment at Duke with Dr. Eddie Smith, and we conduct developmental assessments of the infants at several time points. We do assessments of family and specifically parental functioning and adaptation and caregiver concerns about the child's development. So we have a whole range of battery of assessments that we do for both the parents and the infant over time. In terms of our CKMM assay, this is actually showing our laboratory verification process. We're too new to show this kind of plot for our screening, but I'll show you some of our high-level screening results in a moment. But in terms of our verification, we did our verification process on a little bit over 2,000 infant samples. And based on that data, we set an initial and a final cutoff. There is an important caveat here, and I don't want to get too far in the weeds, but just to mention that because we do our screening after standard newborn screening is done, we actually use slightly older samples. And so that is part of our interest in assessment. And you heard Michelle talk about that as well. CK does degrade over time. So that's something that we're looking carefully at over time. But we have an initial cutoff of 1275 and a second cutoff, a final cutoff of 1626. And at that second cutoff above 1626, that is when an abnormal result would be reported. As of yesterday, we've screened 949 infants. We've had 11 that were incomplete for the reasons shown here. We had 944 that were normal. We have so far had 5 come back as screen positive given our initial cutoff levels, and you can see there are 3 males and 2 females. We did have, we are outside of the kit specifications in terms of the time since the sample was collected. And as I mentioned before, that is something we are closely following given the potential degradation of CKMM over time. And of the 5 positive screening cases we've had so far, remembering that this is very new, so a lot of this is still in process. The first case, a male with a CK of 2400 about, we had a normal latent CK, which is not surprising given the CK value here, and next-gen sequencing is pending. A second male where we also had a normal total latent CK. CKMM actually was insufficient. We didn't have a good enough sample to get that, but the total CK was normal, and also next-gen sequencing is pending. We have an interesting third case where the parents are known carriers for biotinidase deficiency, and that baby is currently being tested. It was not picked up on newborn screening, but is being tested. And they have not consented yet for confirmatory testing and follow-up, given that they want to go through that process first appropriately. We have a fourth case where our latent CK, the specimen was insufficient, and the parents actually declined repeat CK given COVID-related concerns. This actually was last week, and their next-gen sequencing has been sent, is also pending. And then we've just had a fifth case who has not yet been consented for confirmatory testing and follow-up. We just initiated the contact with that family this week. So that is our screening to date. Again, we are very early in this process, but we're very enthusiastic about having this running. This is a large study that has a lot of funding and support. I gratefully acknowledge we have a really incredible expert advisory committee, and several of them are on this call, and it's wonderful to work with them. We also are lucky to be supported by a range of different support, as you can see here. And I will say, specific to CKMM, we are really grateful to Sarepta and to the MDA to help support this work. I'm happy to take any questions.
Thanks, Holly, for that presentation. Sounds like you guys have been busy since you started.
We have.
Does anybody have any questions for Holly? I'm just getting very interesting, cool work, Holly.
Thank you.
So Holly, you do not have a borderline cutoff or option for participants to get a second collection. Is that correct?
Not at this point. It is something that we're considering down the road. Again, once we obtain more data, we have, we are very lucky to have a very nice support and collaboration with PerkinElmer, and they are very interested in doing the next-gen sequencing panel on all of these kids who have increased CK. And at this point, we're quite interested in having that data. It is very likely that we will go to a different approach once we've obtained a larger set of data on early outcomes. So at this point, no, we do not.
Okay. Do you have any other questions for Holly or the other early check team? What's your length of time? So it sounds like you are doing the sequencing from the residual dry blood spot.
Mm-hmm.
Do you have a timeline? This could go to Michelle too, also, of how long it takes for them to get the sequencing results back?
Yeah, that's a great question. We actually don't have the results back on anyone yet. So what we're hearing is anywhere between 4 to 8 weeks. But PerkinElmer has, like I said, been an incredible partner, and they have indicated that they're trying to do ours as quickly as they can, knowing that this is a newborn screening situation. So I will let you know once we start obtaining that data.
Michelle, do you have a timeline for how long it takes you to get the sequencing results?
It's similar. We had delays during COVID, but now they're coming back within a similar time frame, somewhere between 4 to 8 weeks. And some of it depends on, it was being run kind of as a tiered assay. So we've made some tweaks to that, and now the turnaround time seems like it's gotten better. During COVID, it was delays getting the telehealth set up, then delays getting the sample and so on. And so hopefully that'll work out. And again, I agree with Holly. I think going forward, it'll be a more routine kind of a time frame so that we can get those results back, because we don't want to lose parents either and have them be waiting for so long and then dealing with the uncertainty. So.
Well, thank you both. For time purposes, I want to move on and make sure we get May Baker in. So I see May Baker's on the line. May, are you able to share your screen?
Let me try. Okay. Can you see it?
Yep, you can see it.
Okay. Excellent. So, hello, everybody. Sorry, I was a little bit late on the call, and actually, Michelle and Holly's Holly's presentation make my life much, much easier. So I'm going to skip quite a few slides. Let me do this. Okay, you still see everything, right?
Yes.
Okay, let me try. So I just want to adding on a little bit of information about the assay, because our state is not a screening for Duchenne, and second is I think Michelle and Holly has given very comprehensive data, and I just want to make some comments on the assay. So when we had the opportunity to try this assay, and this is before FDA approved, but I do believe it's the similar setting for it. So I think you can take this result, apply to the FDA approved assay. So I will describe the assay, the accuracy, reproducibility, and the population data, and you will find quite a bit similarity as Michelle presented. And the come to assay accuracy, usually in the lab, we have a kind of general principle we call 5 by 5 table. We have 5 replicas, do 5 days, and you can tell this have the range from low, medium, and high. And I am quite pleased with the results, and our staff even make comments if TSH and 72HP have this perform, we'll be very happy. So I think the reason I use that 2 markers to the reference, because they're similar principle, and use the GSP. And you can tell the accuracy, what you expected, and what is, except the lower ones and medium and high, it's all in the 1 SD range. Come to the proficiency, and within round, you can tell, I want to go to very high, guess this through, but I ask you pay attention to the CV, which is a laboratory, very familiar with that. In general, we perhaps accept 15%, but you can tell within days, they are all in 10%. And the between days are even better. So I think this kind of assay give you confidence, and you may be able to do the fixed cutoff. It's just a different slides, go to low, medium, high. And after this is the linearity study. So because it's quantitatively, you want to be sure they are in this linearity, the range. You can tell I have hard time to make slides. What theoretically, what is really we retain. And I really think the assay performed well. And after that, we do some population study. So interesting, kind of, I try to listen to Michelle's presentation, and they did the assay validation, use similar numbers at the end, and about 10,000, and us is here. And this is described the median and in the percentile. And interesting, in this study, this is de-identified. We do have one sample male with a CK 14,674. So you can do the interpretation yourself here. So I think these numbers give you some sense in terms of when you try to think the population, where you really think about your cutoff. Interestingly, we hear Holly talk about they start with 90, then they move up a little bit 95, which is kind of logical because you are conservative until you learn more and you do some adjustment. And in terms of sex, because we do need to talk about that because this is X-linked disorders. And the similar result we see here is male seems to slide a little bit higher than the female. And even this very little, because the sample size, if you do the statistically, they're significant. But as Michelle said, well, that really doesn't matter, doesn't affect your cutoff. And which is kind of makes sense because the female samples, even they have 2 copy X, and one of them should be randomly inactive. So you would expect there the CK level in a similar level, which is the beauty, use the CKMM as a first tier, because if the female have elevated the CK, and there should be justifiable to do the further assessment in case the female have not a random, we call it execute inactivation, and they could have a symptom too. So here is distributed by age. I think, again, we see the similar situation is the earlier you collect samples, the higher your CK. Again, I think it have a biological explanation because the birth is such a event goes to the birth canal, and they have the muscle damage there, and over the time it should be recovered a little bit. And people still think about how about C-section. I thought that if you do C-section, you have to cut. The cut can be this, but I hope this is magical. I know Kathy Svoboda on the call, so she may have a question in terms of agree with this logical explanation or not. But I think we see this, I think seems to make sense. I think it's important we know because how we decided to do our cutoff. So here I showed the figure, and 50%, 95%, and 99% here is the number. So we decided just to compare with the 24, 48, and 96 because they're exact same interval. And indeed, and all this category, 50, 95, 99, and 48 hours compared with 24 hours, you can tell there are CK level reduced by close to, I say 50, a little bit over 50%. And if you compare 96 hours with 48 hours, and it's reduced by 60%. So I think it's all kind of makes sense. So when you decide to cut off, you maybe need to take this consideration. I do believe New York is doing just such. Here is the birth weight. If I heard Michelle, they didn't see much birth weight different. And I think for this figure, you can tell, and in a certain range they do, then diminish. So here is a table showing further. So we use the 1 kilo, 3, and 5 and as a marker to compare. So 3 kilo compares 1 kilo and could have the 4 times higher in the 50%. 95% is a 2.4, and the 99 is further lower. They go to the trend. And 5 kilo compared with 3, you can tell there it's pretty much equal, 0.9, 1, so they're not much. So it's why I said in a certain range, they could be different, but over the time they diminish. So overall, this is just our essay, if you will, call validation, development of validation, and we really think the Perform is to do their job, and we do believe they're qualified good enough to do the first tier, and as the previous two speakers already talked about that, if you really want to know what caused this, the CK elevation, you have to do genetic testing. And when you think about Duchenne, I'm not so sure I use the term CK-MM screening, but if you target the Duchenne, you almost can think about CF. So CF uses the RT as a first tier, and the RT only indicates you have a pancreas problem, but what causes pancreas problem, you need the genetic testing. So this kind of parallel similar situation, but I think, at least I know, for now, I feel RT seems not as stable or as the performs as good as CK-MM. So CK-MM and even program talk about where to set a cutoff placenta. I do believe on the routinely they do have a fixed cutoff of numbers to go about. So here, I'm gonna stop here, take any questions. Thank you.
Thank you, May. That was also a very good presentation and gave us a very good background about things that you need to look out for when you're validating this assay.
Just while other people are thinking of questions, I just want to make a comment. It's Kathy Swoboda to May's comment. So I find it interesting. I think gender has much less to do with variations in CK than potential, African ancestry has been, associated, and the overall size of the baby, as May showed. But what I think probably accounts for the majority of elevated CK is two factors. One, any baby that is monitored during labor, any parent who has seen, they screw this little thing into the baby's scalp while the baby's descending the birth canal to monitor the baby, so that you have this probe, and it goes into the scalp muscle. It actually goes into muscle. So that's one source. The other source, which varies institution to institution, are intramuscular vitamin K injections. I don't know how many programs have moved to doing oral vitamin K. I think most of them still inject it, and that is an intramuscular injection. So I actually think that those two factors outweigh almost anything else in terms of the proximity to birth. That's my hypothesis anyway.
Well, I think I'm glad because with vitamin K injection, I just heard. And what's the consequence when you inject that? You cause the elevation, right? The CK higher.
You're injecting, it's an intramuscular injection. And we know as an EMGer, I'm a neurophysiologist, we know that when you do a CK level after an EMG where you put even a tiny needle into the muscle, mm-hmm. It damages muscle fibers and releases CK.
Gotcha.
So it's really any injection that's intramuscular, depending on a number of issues, can cause leak of CK from that injection site. Basically, that's.
Yeah, your comments on the African American, I think is a very interesting one, because when at least from the data from our laboratory, the explanation we said, because your baby is bigger, you have a more bigger mass. So I think it's a similar thing. I will look into separate them in terms of look at the African American, because even this is de-identified, we did retain all the ethnic the information.
So we have two comment/questions in the chat that I just wanted to interject in. One was, and Holly, if you're still on the call, if you can elaborate, she stated that they're asking parents, I'm guessing they're going to have a survey that outlines the lines that Catherine just asked. Holly, are you still on the?
I am. And so we have a set of questions that we're asking the birth parents. We know they're not going to be excellent reporters of these things. But we are asking them about these different indications of, quote, birth trauma. And the vitamin K injection is a great one that isn't on our list. And we can certainly add that. That would be an interesting question as well. And it's the thing we're not sure of, and it'll be interesting to obtain a set of data and kind of see what we get from parents. But again, we are really quite concerned about how parents are going to be able to report these sorts of data. We have access to APGAR scores, and so those will also be an interesting thing to look at compared to these values.
And then I lost my chat box, but Bob Vogt had asked a question about cesarean C-sections affecting the CKMM value. So is there any evidence that, I'm not sure that's something, that factor that you're collecting, May, that you had in your validation, and I don't know that's a data point that newborn screening programs are collecting, but is there any evidence in that?
So this is me. I can try, and we do not have this information. But if we go in the logical, Kathy was saying, since the C-section, you have a different scenario, I guess. I don't know how you when you cut it and through placenta, how much it goes into the baby, whatever. But you will not have this birth canal situation, and they will not do the injection. But I don't know. That's perhaps still open question there.
Can I just comment again? I would say, it's Kathy Swoboda again. The C-section, again, if it was related, might be, again, the most likely thing that's going to affect the CK more than anything else is screwing that probe in through the baby's scalp. So if a baby's in distress, the first thing they do is they put that probe. Have you seen it? It's a corkscrew. Like you screw it into the baby's muscle in their head. And it's in, and so that can happen during vaginal birth, or if they go to C-section. C-section, you don't hurt the baby ever, really, unless it's a iatrogenic injury where you cut the baby's skin. But I've never heard of a baby, with C-section having muscle cut. I
didn't say well. I think when you go through the surgery, I'm not saying cut babies, like abdomen and open is why I said I don't know the CK will go through the placenta going the baby. So,
oh, I don't think so, because it's
okay.
big.
Yeah.
But I don't know. I guess it's a question, yeah. But the birth canal issue, people always say that, oh, it's so traumatic through the birth canal, et cetera. Whether that really affects muscle leakage of CKMB is MM is completely hypothetical also. And as an aside, as we're doing newborn troponin levels now for gene therapy for SMA, we were originally doing for the Avexis gene therapy trials, the total CK and CKMB, and almost all newborns had, those were elevated too, not just the MM, but also the MB fraction and also the troponin. So, I think there's part of this is, again, establishing the normal values. We're not going to ever figure out all the context of all the different variables, but definitely certain ones that we want to explore should be included in those questionnaires for sure. But parents aren't going to know whether their child, every baby in the US gets vitamin K at birth. That's standard of care. So whether they got it via an injection, I don't remember. I mean, I doubt the parents are going to know that on a questionnaire.
I agree, Kathy. I think you made a very good point. Is as long as you establish the system, you hope, this is a systematic thing, it's not random. Also, I would hope is the infant have a disease, they're much higher. So.
Yeah.
This is Michelle. I remember when our protocol was being reviewed by the IRB, we have a clinician on our IRB who works for the health department, and he was very adamant that breech birth and different types of traumas impact CKMM. So I was interested to hear Kathy say that may or may not be the case.
Is your study collecting any data to understand do you have any demographic data on how the baby was born in your study, Michelle, or no?
Yeah, I was looking through our case assessment and I don't see it on there.
Okay.
I was trying to sort of, that was, is it, because I think he probably brought it up because of that it wasn't on there. And he was, like I said, very adamant that it does impact the values.
Well, I mean, just to comment about breech babies, I mean, babies with muscle disease are more likely to be born breech because they don't position themselves in utero. So, I mean,
It might be related that way, but
it might be related that way, right?
yeah, I mean, it kind of made sense to me, I guess, in a way, but then on the other hand, I was trying to think through it, and certainly a lot of the babies that we have identified, there's very few that have no kind of molecular variant, which I find kind of interesting. So you wonder what the relationship is between all these different factors, whether they have some kind of genetic disease or not, or just they have, because they have markers with either myopathy or neuromuscular type conditions, are they spilling CKMM more because of that? So it's going to be interesting to see how this all fits together.
Yeah, yes. Well, thank you guys for this very interesting discussion. I hate to end it, but we are 10 past the hour. So out of respect for everybody's time, I'm just going to highlight, if you can still see my screen, there's a PerkinElmer virtual summit where there'll be more discussions about Duchenne muscular dystrophy as well as others. We're highlighting these two talks that are particularly on Duchenne, but I believe May, you have and Holly, I believe you guys are on that agenda for that same meeting. So I have the sign up link here, as well as you can always visit the NBSTRN website where there is more information about the meeting that we did a blog post on. And lastly, I wanted to highlight, I'm not sure if someone from APHL is still on the call, but we wanted to highlight their upcoming hot topic webinar, which is on resources and best practices for remote follow-up work. And that's going to be in a couple weeks, December 17th at 2 p.m. Eastern time. So with that said, I'm going to wrap it up. Our next meeting will be in January 7th at 2 p.m. Eastern time. So thanks for everybody for joining. If you have any comments, questions, concerns, feel free to email me. And also everybody stay safe during this time. Thank you.
Thank you.
Thanks, Jennifer.